thank you so much, everybody, for joining us for the second session in the ABC of Genomics series. Today, we're going to talk about the basics of genomics and really try and cover some of those basic terminology and facts that you might need if you're starting to introduce genomics into your clinical practice. So without further ado, just to introduce the ABC Genomics theory series, the idea behind this series was really to keep it as simple as possible, to try and give people a little bit of background information who maybe, for clinicians who maybe haven't done much genomics in their practice to date, or maybe it's been a while since you've done any genomics back in school. So yesterday we did a session about the advances in genomics, which looked at why genomics is happening now and the changes in technology that are driving that. Today we're going to think about, as I said, the basics of genomics, what you do, and ultimately what you don't need to know about genomics in order to introduce it into your practice. And then finally, tomorrow in the third session, we're going to think about clinical pathways and how you introduce genomics within that. So today, as I said, for the basic session, we're going to think about terminology, things such as genomes and chromosomes. We're going to think about how we inherit our genomic information. And finally, we're going to think about types of variants and terminology around that. So I started yesterday's session, session A, with this same slide. So apologies to anybody who's on the same call, but I think it's a really important point to start with, and that's considering what the difference is between genetics and genomics. So a lot of people will have heard of genetics, but less people are familiar with the term genomics. Genomics is a term that we're using increasingly, and it's now the preferred term. So although they often are used interchangeably and do in some ways mean the same thing, genomics is a technically more accurate term, and so that's the reason why we're using it. The difference between the two is that when we refer to genetics, we're talking about the genes themselves, whereas when we talk about genomics, we talk about not just the genes, the coding bit that is coding for those proteins, but we're actually talking about all of the factors that are needed. So the analogy I use is like a book. So in a book, you have chapters of information, and that is what a gene is. It's a chapter of information. But in with that book, we don't just have a chapter and nothing else. We have page numbers, we have a spine to the book, we have binding, we have formatting, we have borders, we have headers, we have footers. We have lots of other factors that enable that book to work to work and to function. And as such, genomics is the same. Genomics talks about all those other functions that might influence how well that book is and how easy it is to use, as well as just the chapters themselves. And so you'll hear in this presentation that I might use those terms interchangeably, and that's okay. And it's okay to maybe use those terms with patients, because patients in particular might be not au fait with that terminology. They might recognise genetics more so. But I will do my best to try and stick to the term genomics, as I say, because it is more technically accurate. So to understand genomics and how we can use it in clinical pathways, we really need to understand the basic principles of cells, chromosomes, DNA, genes and genomes. So let's think through each of these terms and what they mean. So when we refer to genetic in a health or genomics in a healthcare setting, we're referring to a condition which is caused or influenced by genetic factors. These are instructions that we've inherited from our parents and are not necessarily what has been caused by the environment, although I will come on to later in the presentation about how environment can influence our presentation of inherited conditions. The DNA is what encodes for all of the instructions that tell our bodies how to grow and develop, and each instruction is called a gene and is stored in chromosomes. So you can see on the slide that we've got a human body. So I always think about the human body as being made up of bricks that are called cells, almost like a house is made up of bricks, where we have different types of bricks. We have different types of cells. Within the centre of those cells, we have a nucleus, which is where all of our genetic information is stored. Now, we are all probably familiar with the idea that DNA is a double helix or a twisted ladder, like you can see on the screen. And this twisted ladder is tightly condensed into structures called chromosomes, which I often think of as like filing cabinets, which is why there's a set of filing cabinets on the slide in front of you. So if we think about that that storage of genetic information, and we think about a genome and that whole amount of genetic material within our cells. It's, as I said, it's arranged on structures called chromosomes, which essentially are like the filing cabinets. So when we're talking about the genome and a collection of these filing cabinets, essentially we might be thinking about a storeroom. Each single filing cabinet is a chromosome, so containing lots of different genes and genetic factors. Within those chromosomes, we have our genes or our individual files. And as we get down even further into even more detail, we find that the DNA is made up of bases or letters, which are arranged and repeated throughout our genome. And essentially, this is like the text. So we've got our genome, which is our storeroom. We've got our filing cabinets, which are our chromosomes. We've got our genes that are stored within our chromosomes, which are like the files. And within those files, we have our text, which is essentially like the DNA code itself with the nucleotides that we read when we sequence them. So let's just spend a little bit of time about thinking about the different ways that we can inherit genetic information and the way it can be passed on. Firstly, when most people think about inheritance and genetic information being passed on, it's in the context of a parent to child via the sperm and the egg. The cell, or those two cells that come together, the sperm and the egg, form one cell. This then copies and then splits into two, meaning that you then have two cells, and then in turn, those two cells replicate again, so that you then end up with four cells, and so on and so on. So it means that in those cells on the left-hand side of the screen, in a germline, what we call a germline scenario, all the genetic information should be the same in those daughter cells as it is in the parent cell. So if we have a mistake or a spelling mistake or a variant, as we now term it, such as a star, which is shown on that first parent cell on the left, then if we have that mistake from the very start, then what will happen is that all of those cells that result from that initial parent cell will also have the mistake in it. So as I said before, this is referred to as a germline variant, so something that is changing and results from the germ cells themselves, which are the egg and the sperm. Slightly differently, or alternatively, we've got what might be called somatic variants. So this is where you can see on the right-hand side of the screen, we've got the parent cell, which replicates and creates two daughter cells, just like it did on the left. But the variant that has occurred in the somatic inheritance is actually occurring in the daughter cell. So you can see where the star is on the right-hand side of the screen, that actually we've actually got two cells here, one that doesn't have the variant. I don't know if you can see my mouse here moving, but we've got one cell here on the left-hand side of the somatic diagram where it doesn't have a red star, so that doesn't have a variant, and all subsequent cells that come out of that won't have the variant. But on the right-hand side of that diagram, we've got one cell, one daughter cell with a red star, which means that the future daughter cells that come from the replication of this cell will also have that variant. So if we look on this bottom line of the somatic cell, we've actually ended up with different types of cells. We've got some cells with the variant in on the right hand side, and we've got some without. And this type of inheritance is known as somatic and can often end up with what we call a mosaic pattern, because somebody is a mosaic, is ending up with different types of cells. But for the purpose of this presentation, for the next few slides, we're going to focus on just this germline inheritance. So somatic inheritance is often the case in cancer, where you get a cancer change in a particular cell. But for a lot of genetic inheritance, where we're talking about things passed on from generation to generation, it's germline inheritance that we're talking about. So that's the bit that we're going to focus on in the next few slides. So to understand inheritance, we need to first understand a little bit more about those chromosomes, or those filing cabinets that I've just been talking about. We need to know about where they come from and how many we have. So in a typical human, there are 46 chromosomes, which are arranged into 23 pairs. So the picture on the screen is a photograph taken down a powerful microscope after a cell has been stained, which shows the chromosomes. So you can see here that we have chromosome 1 on the left-hand side at the top, And you can see that these are the biggest chromosomes. They don't look like they match, but people in the lab, such as cytogeneticists, are skilled enough to recognise which of the chromosomes are the same. They don't neatly line up in a straight line, because if you remember, these are very tightly coiled and squashed into the nucleus in the middle of the cell. So when we take a photograph of them, they're not necessarily completely straight as sometimes you see in computer illustrations, but rather they're these different structures. They look a little bit like worms when you see a picture where it hasn't been segregated. And this isn't just a photograph that's taken from a cell like this. This has been manipulated and worked. So the people in the lab have been able to take a photograph, chop it up and rearrange it so that they can see the chromosomes in order. And you'll see that on the left-hand side, as I say, chromosome number 1 is the biggest. Right the way down to at the bottom, we've got chromosome 22, which is the smallest. But you'll actually see that next to chromosome number 22, we've got the sex chromosome. So these are the 23rd pair. And here this person has got an X and a Y, which means that they are male. So we have pairs of chromosomes because we inherit one copy from our mother and we inherit one copy from our father. So the eggs and the sperm only have one copy of each chromosome in them, because when they come together, they make a double set, which is a set that the child has inherited. So in turn, when we have our own children, we will pass on just one copy of each chromosome, because they will in fact get another set of chromosomes from their biological parents. And it's this simple fact that underlies a lot of how genetic conditions are passed on and the risk to patients or the risk to children. So I want to go on now to just think about the 3 main types of inheritance. So I just want to give credit to Illumina here, who created a counselling guide for reproductive genetics, and it's available via it's a free resource, and it's available via the QR code in the bottom right hand of the screen. But these diagrams are taken from there, and I just wanted to give credit to that. So if we consider typical inheritance patterns of germline, I think this is often where people reflect that they find it very confusing with genetics. But essentially, there are 3 main patterns to inheritance. There is dominant, there is recessive, and there is sex-linked. There are a few others, but they are very rare. And generally, if they were to be present in a clinical case, then it would be worthwhile involving clinical genetics to help with the management of that. So firstly, dominant inheritance then. So again, we're talking about the idea that we inherit one copy of the gene and one copy of the chromosome from our mothers and one from our fathers. And in turn, we're passing one copy to our children. In dominant conditions, if you have a variant in one particular copy of your gene, then it is sufficient to be dominant in effect over the other copy. So that's why we say it's dominant. So you only need to inherit one copy in order to be at risk of developing the condition. And if we have a look at this in terms of what's on the slide in front of us, what we can see is that here we have a father who is carrying the variant, or the mutation as it's labelled on the slide. He has one copy of the variant and one normal copy, whilst the mother has 2 normal copies. When that father comes to have his children, he will pass on half in his sperm will have the copy with the variant on, and half of his sperm will have the copy without the variant on, which means it's completely potluck which sperm is used to fertilise the egg and which copy the child inherits. So if we look at the 4 possible options of outcomes for his children, so in the first scenario, the first child inherits, and in this scenario, although they're shown as male or female as the children at the bottom, it really doesn't matter because it's not a sex-linked inheritance. So we can ignore what sex they are, more what colour they are is what we're looking at. So on the left-hand side, we've got a child who's inherited a copy of the variant from his father, but a normal copy from his mother. In the second scenario here, we've got a daughter who again has inherited the variant from his father, but has inherited the other slightly darker copy of the normal copy from their mother. And similarly, we've got another child here who's inherited a normal copy from the father and a normal copy from the mother, so are not affected. And again, the 4th child has inherited the normal copy from the father and the other normal copy from the mother. So the pedigree that's on the right-hand side shows a typical pedigree, that how it might present in clinic, where we have multiple generations of the family who are affected with the condition. We have males and females affected, because again, it's not sex-linked, and we see all forms of transmission, including from male to male transmission, and that's key when we come on to think about sex inheritance, which I'll come to in a moment. For recessive inheritance, this time inheriting one copy of the variant doesn't necessarily mean that that person is at risk of developing the condition. That's because the second copy, the second normal copy without the variant, is sufficient to be able to compensate for the lack of the non-working copy. So again, in this scenario, if we look at the diagram on the left-hand side of the screen, we can see that the father has got both a variant and a normal copy, and the mother also has a variant and a normal copy, which means that the combination for their children is that the first child on the left could inherit the variant from the father and the variant from the mother. Now, in this scenario where they've inherited the two variants, they then don't have a copy to compensate, so they're therefore at risk of developing the condition. In a second scenario, we have the father has passed on the variant to the child, but the mother has passed on their normal copy. So this person here would be known as a carrier, because they carry the variant, but they themselves are healthy. And in a similar scenario, but the other way around, this child here has inherited the normal copy from the father, but the variant from their mother. Again, they are carriers. Or in the final scenario, we have the normal copy passed on from the father and the normal copy from the mother. So in this scenario, this child hasn't inherited anything and is not at risk of passing it on. So if we look at the ratios that we can see out of the children at the bottom, we've got a 1 in 4 chance that the child is affected. We've got a 2 in 4 or a 1 in 2 or a 50% chance that the child is a carrier. And we've got a 1 in 4 chance that the child is unaffected. And again, if we look at this in the context of a family history, we can see that very often in recessive conditions, because people in the family are carriers but themselves not are unaffected, we can often see that there isn't any evidence of the genetic condition in the previous generations, and that it might have occurred for the first time in the children. With very rare genetic conditions, these sometimes become more prevalent in couples where there is consanguinity, because the variant is so rare, but because there's intermarriage relations, it means that it increases the risk of them sharing some of those very rare genetic factors. Sometimes there is evidence higher up in the family history. For example, CF is a very common condition in our population, and so therefore it may well be that you have others in the wider family with CF. So you do sometimes see it, but more often than not, it's occurring for the first time in that generation, but it can occur more than once, because once there are 2 parent carriers, that risk of having an affected child goes up to 1 in 4. So siblings can often be affected, as can males and females. So the final type of inheritance that we're going to look at in a bit more detail is X-linked inheritance. And this it becomes a little bit more complicated because of what we talked about earlier with the chromosomes and thinking about the X and the Y. So for females, we have 2 X's, and for males, we have an X and a Y. So that means that if we have a inherited tendency or inherited condition which is existing on the X chromosome, if you are a female and have a variant on your X chromosome, essentially this works like a recessive condition, because although you carry the variant, you have a second copy of the X chromosome that is going to compensate. Whereas if you are a male and already have a Y chromosome and then inherit an X chromosome with a variant on it, it will mean that you don't then have a working copy of that gene. So for males, it can look like it's dominant. So if we look at the pattern, if we think about that in the context of how that might be inherited, and again, look at the pattern on the screen, we can have males. This is where it does matter whether the children are male or female. We can have a male child who has inherited the Y chromosome from the father and the normal X chromosome from the mother. So in which case, it means that this son, this male child here, isn't at risk of developing the condition. Similarly with a female, they've inherited, this female here has inherited a working X copy from the father and a working X copy from the mother. So there again, they're unaffected and there is no risk of them being able to pass this on to their children. If we look again at the third option, the father is again passed on the normal X chromosome, but this time the mother has passed on the Y chromosome, the affected X chromosome, which again, because there's two X's, means that this person would be female, but it will also mean that they are at risk of passing this on. So they've got two copies, one with a variant in, so they've got one to compensate, so they're not at risk of developing the condition, but they're at risk of passing this on, and that would be particularly key for sons. And then the final option is that there's an affected son in that they have inherited a Y chromosome from their father, but they've inherited the affected variant copy from their mother. They don't then have that extra copy to compensate, so they're then at risk of being affected by those symptoms. And again, if we look at the ratio that gives us, it means that for every pregnancy, there is a 1 in 4 risk of having an affected son. There is a 1 in 4 risk of having an affected daughter, and of having a carrier daughter, and there is a 50% chance of having a child, male or female, who is neither affected nor a carrier. If we look at this in the context of a family history, what we can see is that there is often more than one generation involved, and it's only ever males that are affected usually, although you can sometimes usually see very mildly affected females, but I'm not going to go into too much detail of the reasons for that here. And also you'll never see male-to-male transmission. In terms of other genetic modes of inheritance, they do exist. We have mitochondrial, we have mosaic, and we have multifactorial. But I'm not going to go into those in a massive detail. Again, if you do come across those in your clinical practice, then it's probably worthwhile reaching out to clinical genetics who'll be able to advise those. But just before we do move on, I just want to talk a little bit about polygenetics and epigenetics. Polygenetics, poly means multiple and genetics obviously means inherited. So polygenetics refers to scenarios where there might be multiple genetic factors which are affecting the inheritance rather than just these single factors as we've been talking through. And often I think where we get negative results but we get family clustering, this is possibly part of the reason. With epigenetics, it's a term that is increasingly popping up, and it refers to factors that are influencing how our genome is regulated without it actually being the DNA code itself that's being changed. So if you look at the image on the right hand side, thinking again about those filing cabinets, with epigenetics, we're thinking about the factors which might influence whether those drawers are opened and whether those files are accessed. So there can be certain factors, environmental and genetic factors, which can influence that, and we're realizing that those epigenetic factors can really influence somebody's presentation. Some of the epigenetic factors are listed on the screen. So environmental factors like the medication that we take, the pollution and environment that we're in, how much exercise we take, our diets, things like smoking and alcohol, all affect those epigenetics and can affect whether we present or not. And it's often these that then can help confuse the matter as to whether something is genetic or not. And one thing to remember is that often families will share the same environment, certainly amongst childhood, and it might be shared environmental factors that are influencing the family clustering rather than specific genetic factors that are being passed on, like we've discussed already. So key tips and tricks for spotting a genetic condition. We need to think the red flags, think about the number of people that are affected within the family with the same or similar condition, the age at which people are presenting in genetic conditions. This is often younger than you would normally see in the general population. And also think about the types. Are they clustering and are they patterns that we might recognise often go together? So what do we actually call it? So I've been using the term variant throughout this presentation, but in fact, there are lots of ways of talking about a change in the genetic code. We might refer to them as a spelling mistake, a mutant, a change, a fault, an alteration, a mutation, a variant, or an error. But by far, the most popular term is variant, and that is what you will see written on your genetic reports. On the slide is an actual copy of a sequence of DNA, and this is how we see them in the lab and how we're read. And the eagle-eyed amongst you will be able to spot that there's a difference between the reference section that we have at the top and the patient's sample at the bottom. And what's happened is that the patient has actually got an extra bit of DNA where it's missing from the reference section. They've actually got an extra bit of DNA. And if you look even closer, you'll see that this is actually a repeat of this sequence just before it. So we know that they've got a small duplication in their sequence. But the question is whether that makes a difference or not for the patient. So lots of us carry these changes in our DNA. Many of them are benign and don't cause a problem. And we can still refer to them as variants. So we know that this can be confusing, particularly in practice for talking to patients about it. So it's really important that when you are reading the reports that you understand the distinction between these three terms on the screen. So the first type of variant that we describe is a pathogenic variant. And when we're talking about something being pathogenic, we're talking about the fact that it causes disease. So it is known to be disrupting the function of that gene, and it's likely to cause issues with the family and is likely to be the underlying cause of their presentation. The second type of report that you might see is a no variants detected. That means that there's no change in the sequence being picked up at this point in time, or none that we recognise that would be a problem and cause an issue to the person's health. And the final type of variant might be described as a variant of unknown significance. And this is where we've got a change in the code, but we're very unclear as to whether this is disease causing or not. And unfortunately, when we find a variant of unknown significance, we have to treat it like a negative result. So we wouldn't use that for any cascade testing or testing within the family to establish risk. It would simply be noted, and that would be reviewed every few years or so to see whether through research and through extended testing in other areas, we've been able to define that variant any further. So to summarise then, what do you need to know? So in mainstream genetics, you need to know the types of red flags that might indicate that there might be something genetic going on in a patient. We need to understand what genomics can do for patients and understand what it can offer in terms of clarification and establishing risk for relatives. And we need to know what the terms gene, chromosome, variant, a pathogenic, VUS, and non-detected mean, so that when we see a report, we can help to interpret that in the context of the patient. What you don't need to know is all the complex gene names that exist. You don't need to know about complex patterns of inheritance, and you certainly don't need to know how to cascade information to relatives or how to test unaffected patients, because for those bottom two, clinical genetics still exist, and that is still very much their role. So I hope you found the session helpful. I'm going to stop the slides in a moment, but if you are engaged and feel like you would like to know more, then you can follow us on social media and through the ways shown on the slide, or you can sign up to our newsletter with the QR code at the top. You might choose to do some more education, so you might want to join us for the third and final series tomorrow, or you might want to have a look at some of our previous Lunch and Learns, which go over some of the topics that we've talked about today. You also might want to check out Genotes, which is a resource for clinicians, which has both an encyclopedia of knowledge and also a in-clinic resource. And finally, you might want to check out some formal learning either through the links on our website or through the Genomics Education Programme website, again, whose QR code and web link is shown on the slide. And they have a huge offer of resources and courses, from quick 2-minute videos and images right the way through to master's programmes. So I really hope this has been helpful. Feel free to fill out our evaluation form if you want to give us some feedback about what you have or haven't enjoyed. We'd be really grateful for all honest comments and feedback. The evaluation form is also linked to our certificate of attendance. So you would, if you'd like for your CPD to receive some certificate of attendance, then please feel free to fill out the form. The form itself literally takes no more than about 2 minutes to fill out, so shouldn't take too much of your time. So I'm just going to stop the slides now. I will put the link for that evaluation form into the chat, but I'm happy to take any questions at all. So I tried to keep it short and sweet today, because obviously I'm conscious that clinical time is just so precious. But there are lots and lots of different resources. If you found it confusing what I've discussed today or shared, then please feel free to reach out to me. Feel free to say if there was something that was confusing from the presentation. Feel free to ask me to go over anything again. That's no problem at all.